Hi, good morning, and welcome to the ZP Vlog and Podcast. We do this every Sunday at 8 a.m. London time, and we really just do a wrap up on the sort of the news from ZP for this week. So if I essentially jump into it, then some of the news from this week, we put out a little bit of a note on the ZP organizational structure. This might seem a little sort of unusual, but it really sort of says that a company like ZP, companies sometimes structure themselves very rigidly into departments, into teams, and even into functions. So you can kind of have like a marketing department and a R&D department, for example, and people very specifically, are doing marketing or doing R&D in those departments. With a smaller business, people can have many hats, many functions, and actually and are, genuinely multi-skilled. So at ZP, we sort of run a matrix organization, and we do try to, define a matrix organization as, in some ways, people can work in different departments, go across different departments, go across different functions. And what you notice then is, if somebody's got a budget to have what we would call five full-time equivalents on their project, then what ends up happening is we can end up bringing about 32 people to that actual project. So they will get somebody who's excellent at electronics, somebody who's got very fine sort of soldering skills, who's good at 3D printing, somebody who's very good at functionalization of electrodes, somebody else who's very good at um, manufacturing those electrodes. So we do run a matrix organization. This just basically means that we try to be more flexible than a kind of very hierarchical structure, and it does actually, bring benefits to us. But I suppose more importantly, it actually brings benefits to the client programs as well. So we did put some news out about that this week. So almost at the heartbeat of what we do towards the sort of biosensor development community is our weekly developer zone. So this week, we dealt with questions which were on cell culture monitoring and bioreactors, screen printed electrodes for protein detection, Faradaic impedance, ammonium sensors, applications of ZP, CGM, continuous glucose monitoring. We touched on cortisol sensing. We talked about chloride resilient pH sensors. We talked about sensors for bioprocessing. And we also talked about sensors for nutrients as well. So it's just to say that if you have any technical questions of the ZP, then we do this Developer Zone webinar every Thursday at 8 a.m. London time. And I think it really helps people. They got a sort of question, they're stumbling over it. We had one guy who's got some CGM or continuous glucose monitoring products from us. And, he was essentially listening live and he was able to give me real time feedback. And I was actually responding to him as he was asking his questions. So it does demonstrate that those webinars are, not rehearsed, they're not rigid. We're able to flow with the feedback in real time as people speak to us, essentially. Something else that we put out this week was actually about EIS, electrochemical impedance spectroscopy. But I realized that this is actually bigger than just impedance spectroscopy. One of the things that we do at ZP, um, it's it should be the standard, but maybe it's not necessarily the standard. But maybe this is something I will work on with my academic colleagues and when I go to academic conferences is large scale testing. I say on the blog this week, large scale EIS testing, electrochemical impedance testing. But I realize that when you read scientific papers, um, then you'll have a signal versus concentration. But sometimes those signals are sort of one test at each concentration. The stronger papers, it's triplicate. But I realize that when we do work, we do really large numbers. And so, for example, we were doing some work this week in EIS. I happened to be in the laboratory at the time, and there were two scientists, um, one of them making the samples and the other one doing the testing. Um, Now, that's the luxury of being a business, that the fact is we can do that. Now, I understand that during a PhD, etc., it might actually only be one scientist. So you've got a scientist that's both making the solutions, and and the same scientist is doing the testing. But we industrialize testing of electrochemistry and the testing of electrochemical biosensors. We really industrialized this. So we have two scientists, one making, one testing, and they tested. I counted the tests or the samples up. They tested 90 samples. So when we're trying to make a consideration, in this case it was impedance spectroscopy, what's the impedance spectroscopy signal versus concentrations? We did it on 90 tests. And I just don't think necessarily that kind of testing is happening in electrochemistry. And I realize at the moment that in biology, they actually do testing in 96 wells quite routinely. But it's just not done so routinely in biosensing. So maybe that'll be one of the things we really want to work on. Actually, is the idea that if 96 well plate testing is the sort of default in biology, why is it not the default testing in, let's say, electrochemical biosensors? And I think the answer is it should be the default sort of number of testing. Something else that we did this week, which was really quite, it's really part of a series now that we've really sort of established, is workshops. So at Zimmer Peacock, there's many ways of learning with ZP. We have our ZP Academy. We give internships. We have these YouTube channels which are have a lot of really quite good material on them. But then I think almost the best way and the most powerful way is actually our workshops. And so this workshop, we held a workshop in Swansea University. It was really well attended, and I think people came away from it and really gave really positive feedback. And it was just some things are quite straightforward, like the connection between cyclovoltammetry and amperometry. We just taught the fact that in order to do an amperometric experiment, you're probably best off doing a cyclic voltammetry experiment first. And so the workshop was really good. We had some people from industry, some people from academia, and the blend between, we talk about the commercial aspects of biosensing, the theoretical aspects, but then we sort of wrap it all together because actually we have a very strong I would say sort of testing aspect to it. Everyone goes into the lab, everyone makes a biosensor, and everyone gets to test a biosensor. So I think people were really super intrigued by the whole experience and gave some very positive feedback. At ZP as well, we do have some very nice collaborations. So one of our collaborators at the moment is developing a very nice aflatoxin B1 sensor. And so we just put a little note out about aflatoxin B1. It's really interesting that this is really a kind of carcinogenic chemical that's put up by fungus. Aspergilla flavus is the culprit in this case, and this kind of gets onto a lot of organic materials, a lot of sort of farm-produced materials, and it can be toxic both to humans, but to cattle, etc. And so we just put a little article about this molecule, and that look forward to really some rapid tests coming around aflatoxin as well. And I think I just made the point that actually we have this kind of sense it all platform at ZP, and the word sense it all is not just sort of spurious or frivolous comments. It can actually test aflatoxin B1s as well. And then finally, the last bit of news this week is ZP. We have a really nice product. It's called the Food Sense. It could do full food quality testing, including measuring the hotness of chili sauces, chili products. And I just wanted to kind of put a little video out there this week, really talking about the solubility of capsaicin. It's really interesting, actually. You can make a point of need test. I saw it called a POCT, a point of care test, this week. So you can take this kind of chili sauce, you can prepare a sample, you can put it on the sensor, and you can get a result. Great, we're all super happy. But but capsaicin is kind of interesting because it's got a limited solubility, and in fact, I think I say in the video that it's kind of got a solubility that runs roughly from 13 ppm, 13 parts per million, to about 29 ppm. ppm is milligrams per liter. It's a useful term. Lots of engineers use it. I don't think absolutely everyone actually completely understands what it is, but it gives them a sort of units to put. So we were saying that when we were testing the bomb is a very pungent, hot, let's say, sauce. We were actually doing a 1 in 500 dilution, and that's because the natural solubility of capsaicin in sort of waters and buffers is about 13 ppm, which is 208 Scoville units, to about 29 ppm, which is 433 Scoville heat units. So when you're trying to measure something that's advertising itself as like 100,000 Scoville heat units, we have to do quite high dilutions. And I just wanted to kind of give a rationale as to why sometimes we are, for example, testing Tabasco sauce, that's 1 in 4. Why is that low relative to the idea of 1 in 500 for the bomb? And it's because Tabasco's got a hotness of 2,500 Scoville units, whilst the bomb, as I say, is advertised at, I think on the side of the bottle it says about 135,000 Scoville units. So it's just that difference in apparent hotness that means that we have to use more buffer with the hotter solution. So we just did a video to summarize that. So we've put out a little bit of news this week. It's fair to say it's always kind of eclectic that we talked about ourselves in terms of our structure and how our structure can make us efficient. We've talked about specific client problems and how people can engage with us. We've talked about how we're doing a lot higher throughput testing than most companies and most groups do. So, for example, just one EIS experiment, involved 2 scientists and 90 samples were tested. Of course, as part of our kind of mindset and mantra, we do train other people, and of course that Swansea University is a good example of that. So in March, we definitely have workshops online, workshops in Norway, and workshops in the UK. And then we're really looking forward to one of our collaborators who's bringing out an aflatoxin sensor soon, so we do look forward to that. And I think this is technically an interesting comment for people who are developing biosensors, that sometimes you kind of run into unusual issues, and one of them could be the solubility of your compound of interest. And so I've given you essentially a strategy on how to overcome that. If you have any questions on Zimmer Peacock, reach out to us. We will do our webinar every Thursday at 8 AM London time, where we answer more technical questions. But that's really the news from ZP for this week. I wish you a good week, and I shall speak to some of you on Thursday. Okay, thanks very much.